where we left off last week was sort of finishing up with protein synthesis, talking about nucleic acids and how they direct the creation of proteins. And so we'll just kind of summarize that here in the slide. I know we already did this, but let's just kind of make sure we're understanding. So we were talking about nucleic acids, one of the large polymer molecules that makes up our DNA. Huge molecules with millions of pieces connected. And the building blocks of those nucleic acids were these nucleotides. And a single nucleotide has three parts: a phosphate, a sugar, and a base. How many different bases are there? 4. And the sequence of those 4 bases, that's our genetic code. That determines who we are, how our body functions, how our body looks. Okay? All those details about how to build you are stored in your DNA in the sequence of A's, T's, G's, and C's. So the genetic code we know is universal. Okay? All organisms of all types have the same genetic code. We have about 3 billion pairs of these bases throughout our entire genome, our entire DNA. And so what the DNA really does is it has the recipe, has the information about how to build all of the different proteins that make up our body. Remember, we're basically protein. And so which proteins are made, when they are made, determines how living things function. And that's stored in the DNA bases. So you need DNA has the recipe, but it's stuck inside the nucleus. It's kind of protected. So our chromosomes are in the nucleus, but they don't leave the nucleus. So in our DNA are these recipes about how to build various proteins, but the recipe's stuck there in the nucleus. In order to actually get that protein made, a copy of that recipe needs to be made first. And the molecule that does that is messenger RNA. It's a type of RNA, which is another nucleic acid, that basically copies down the genes, and then it can leave the nucleus. So the DNA opens up, like you see here. The gene, which is a section of the DNA, gets copied to messenger RNA, which then can leave the nucleus and go to the ribosome. The ribosome is also made of RNA, a type of RNA we call rRNA. So the messenger RNA sort of attaches to the ribosome, and the ribosome helps put the different amino acids together. A different type of RNA, tRNA, transfer RNA, actually brings in the correct amino acids. Okay? It sort of has bases in it that match up with the messenger RNA. It says, okay, we need a leucine, we need an arginine, we need a glutamic acid. And so each transfer RNA brings in the correct amino acid, and they sort of get linked together at the ribosome. And so you have this sort of string of beads that is being connected. Each of these blue circles is one of the amino acids. So it's starting to make a polypeptide as the transfer RNA brings in the correct amino acids. This happens, to build the entire protein hundreds of times or thousands of times, connecting these amino acids together until the protein's finally done. There's a special sequence of bases in the messenger RNA that tells the ribosome when the protein is finished. It's called the stop codon. And then the protein detaches, and it's been made. It's been made with a specific sequence of amino acids, kind of folds up into its special shape, and it's done. So the steps of protein synthesis they have 2 different names. One is transcription. Transcription is when you take the DNA gene and copy it to messenger RNA. Happens inside the nucleus. And the way it works Makes sense. So you have DNA, which is typically this coiled up double helix. But when it's going to be transcribed into messenger RNA, it kind of unwinds partially. Doesn't completely unzip from one end to the other. The area that's going to be copied is. The enzyme that helps that happen is called RNA polymerase, which makes sense. Polymerase means to build up a polymer. It's building up an RNA polymer based on the bases that are in this DNA. And so the RNA strand is created. So if we look, let's pretend that this DNA, the bases that were found here are G, G, C, A, T, A, C, C, G, A, T. Okay. That's starting to make the recipe to build this protein. What would the matching messenger RNA be? What goes with G? C. What goes with G? C goes with? A goes with? No. Doesn't go with T. Remember, RNA has a difference. It doesn't have T. What does it have? U. Uracil instead of thymine. And so these messenger RNA bases are complementary to the DNA bases. C's with G's. Okay? T goes with A still, but A goes with U when you're making messenger RNA. Where's this messenger RNA headed? To the ribosome. And when it gets to the ribosome, it's ready for what we call translation, which is actually building the protein. So transcription is to transcribe the DNA gene into messenger RNA. Translation is to take that recipe and then actually build the protein. So the way that the genetic code works, every 3 nucleotides, every 3 bases is a code. A code for one amino acid. We call that a codon. Okay? So for example, here's the messenger RNA bases. G, U, C, C, A, G, G, A, G. Every 3 is one codon. Okay. So this is a transfer RNA molecule that's going to be bringing in the proper amino acids. It reads GAG. It has matching anticodons that the transfer RNA can attach to bring in the correct amino acid. Okay? And so what's the next codon that will be after this one? CCA. So another transfer RNA would bring in the right amino acid for CCA, and they'd be linked together. There's special codons. One that says start, that sort of begins the process, and then there's a special stop codon, which means the protein is finished. So you have a chart, and we'll get into this chart more when we get to our genetics unit, but this chart is the universal genetic code. All organisms have the same genetic code. And what it does is it tells you which codons are codes for which amino acid. For example, if the messenger RNA has the codon UUU, that is a code for an amino acid, a phenylalanine. If it has GAG, that's glutamic acid. If the codon is UGA, that's the stop codon, which means when the messenger RNA, when the transfer RNA and the ribosome reach that, it means, okay, protein finished, let it go, we made the protein correctly. So this chart identifies which codons code for which amino acids. And finally, just to summarize again, DNA gets transcribed to messenger RNA, which goes to the ribosome, where transfer RNAs bring in the correct amino acids according to the recipe. They get linked together until the protein is finished. That's how the cells make proteins, by reading the code in the DNA, changing it to messenger RNA, and using that to build up the protein. Any questions about that? All right, so that's the end of our notes on the biochemistry portion of this unit.